Welcome friends, today we will start a chapter photosynthesis from botany course. According to the syllabus, it comes under the lecture number 7, 8 and 9. In this chapter photosynthesis there are 10 main points. The first one is a definition and the equation. In this point we will see the definition and the process and the overall equation of the photosynthesis. The second one is history. In this point we will see the various contribution of the scientist on photosynthesis. Third one is the structure of the chloroplast. It is very important. We will see the ultrastructure of the chloroplast and various components of the chloroplast. The fourth one is photosynthetic pigments. There are various photosynthetic pigments like chlorophyll, carotenoids and phycobilins and various we'll see next. And the fifth one is the light reaction. We'll see the in this point the photophosphorylation and the types of photophosphorylation. And the sixth one is dark reaction. There are three pathways in the dark reaction. We'll see next, and this is very vast one. And seventh one is it is new concept one, immersion effect. And the eighth one is photorespiration. And the ninth one is interdependence of the light and dark reaction. And the last one is factors affecting on the photosynthesis. In this point there are two factors. One is external factors and the second one is internal factors. So we'll start from the definition and the equation. See the definition of the photosynthesis. It is large but a simple definition. See, it is an intracellular anabolic process in which glucose is synthesized in the presence of light and chlorophyll A using simple inorganic substances like carbon dioxide and water, with the evolution of oxygen as a byproduct. And the photosynthesis is a redox reaction, means in the photosynthesis there are both the reactions takes place, that is oxidation and reduction. And the second one is the photosynthesis is a anabolic process, means in photosynthesis the solar energy is used for the synthesis of a complex molecule glucose from simple forms. Means anabolic process means synthesis. And the other one is catabolic process. The respiration comes under catabolic process and the photosynthesis comes under anabolic process. And the third one is the photosynthesis is a intergonic process. It is new one. Intergonic process requires absorption of energy, and the sunlight provides necessary energy to plants for the production of sugars during photosynthesis. Hence the photosynthesis is a intergonic process. We'll see in this is the photosynthesis, this is word equation, and this is the simple equation, symbol equation. Carbon dioxide and water gets converted into glucose and oxygen by using the light and chlorophyll. And here the symbol equation, 6 CO2 molecules combined with 6 water molecules in the presence of light and chlorophyll, the formation of glucose and release of oxygen takes place. The oxygen is released in the atmosphere and glucose is stored in the leaves. So this is the photosynthesis. Roots gets absorb water from the soil, and leaves takes carbon dioxide from atmosphere and takes sunlight, and the photosynthesis takes place in a plant, and oxygen get released in the atmosphere and sugar in the form of glucose is stored in the leaves. Move to the next. Second one is history. First, the various different scientists. The first one is Van Helmont in 1684. He observed the willow tree which is grown in weighted amount of soil. He proposed that Dry weight of plant increases due to H2O and soil. This is the experiment of an helmet. Day first, the weight of tree is 5 IB tree, and after 5 years later, the weight of tree is 169 IB tree. This is the Van Helmont experiment. The weight of dry tree increases. Go to the next. The Woodward. He tested the Van Helmont hypothesis. And next one is the Stephen Hales. It was very important. He was the father of plant physiology. Stephen Hales was the father of plant physiology. He first explains the relationship between the sunlight and the leaves, and he discovered the green plants require sunlight for their nourishment. And spelling mistake. So go to the next. Joseph Priestley in 1772. He is famous for his bell jar experiment. He first discovered the oxygen, also known as he was the father of evolution of oxygen, and he gave an idea gas exchange taking place in the photosynthesis. See the bell jar experiment. The first one is here the mouse will be dies, and after putting a small tree in the jar, the mouse will be lives and the candle will be. So this is the Joseph Priestley bell jar experiment. Next one is Ingenhouse. In 1779, he recognized the participation of chlorophyll and light in the process of photosynthesis. Also, he discovered of the plant gives oxygen in the presence of light and CO2 in the absence of light. The next, Jean Senebier. He proposed in the presence of light, green plants absorb CO2 and release O2. Next one is Nicholas Desaugiers. He gave the importance of water in the process of photosynthesis. Eighth one is the Pelletier and Cavington. In 1845, he discovered and named the green pigment chlorophyll. Who discovered the chlorophyll? Pelletier and Cavington in 1845. Next one is J.R. Mayer. In 1845, he proposed green plants utilize light energy and converts it into chemical energy of organic matter. To the next one, Sachs et al. He demonstrated that the first visible product of photosynthesis and glucose is stored in the form of starch. The very nice contribution of this Sachs et al. The first visible product of photosynthesis he gave first. The next one is Eagleman. He plotted the action spectrum of photosynthesis. His materials for this experiment: prism, illuminated cladophora, suspension of aerobic bacteria, and accumulation of bacteria in the region of blue and red region. Next please. The accumulation of bacteria in the blue and red region of light. See the chlorophyll B and the chlorophyll A, red and blue region. Action. This is the action spectrum of photosynthesis. We'll get to the next. F.F. Blackman in 1905. He discovered the two steps in photosynthesis. First one is light reaction and dark reaction, and he proposed the law of limiting factor. So we'll see the law of limiting factor. He states that when a process is conditioned as to its rapidly by a number of separate factors, The rate of process is limited by the pace of the slowest factor. It is because the low light intensity has become a limiting factor. This is the law of limiting factor. Let's move to the next. Warburg. He first used chlorella to study photosynthesis. The chlorella, the different chlorella used to study the photosynthesis, like chlorophyta, chlorellus, and Tribioxophyceae. And chlorella, the first alga to be isolated in culture by Benjering in 1890. The Van Niel in 1935, he found that some bacteria use H2S instead of H2O in photosynthesis. Uh, some bacteria use hydrogen sulfide instead of water in the photosynthesis. He first scientist to demonstrate the photosynthesis is light dependent redox reaction, and in which H2 from an oxidizable compound reduces CO2 to cellular materials. He expressed as the 2H2O plus CO2 and gets converted into 2A plus CH2O plus H2O. This is the, and next we'll say the Emerson and Arnold in 1935. They stated that light reaction consists of two distinct photochemical processes. Robert Hill, this is one important one. He discovered the production of O2, oxygen, without acceptance of carbon dioxide fixation. He discovered the photolysis of water, which is also called as Hill's reaction. The Hill's reagent, he used the reagent one, that is known as Hill's reagent, 2,6-DC-PIP, means 2,6-dichloro phenol indophenol. And the next one is Ruben, Hasid and Kamen in 1941. They proved oxygen evolved in the photosynthesis comes from water. And the second last one, Melvin Kelvin, one most important one is in 1954 he confirmed the fixation of CO2, that is Kelvin or C3 cycle. He's also proposed that the first stable product is PGA, means phosphoglyceric acid, and he got a Nobel Prize for this in 1961. The last one is Hatch and Slack in 1967. He confirmed a C4 pathway for fixation of CO2 in tropical grasses. We'll see the contributions of various scientists. We'll see next the structure of the chloroplast. This is the chloroplast. This is the leaves, and in this leaves the mesophyll leaf cross section. And this is single mesophyll cells. In these mesophyll cells, the chloroplasts are present. This is the chloroplast. This is stroma. This is granum. This is thylakoids. This is the outer membrane. This is the inner membrane. These between outer membrane and inner membrane, the intermembrane space is present. Move to the next. This is the structure of chloroplast. First discovered the Kemper. The chloroplast in the green plants are the site of photosynthesis. This is the lens shaped, and this is the outer membrane. This is the inner membrane. This is middle periplastidial space contained with periplastidial fluid, and this is double membrane called peristromium. It is made up of lipoprotein. The size is 4 to 6 nanometer. Next, internal part of the chloroplast. These are the thylakoids. It is then made the thylakoid membrane and forms grana lamellae, 5 to 25 grana lamellae in each grana, and it is granum, and granum is made up of 40 to 60 grana. It is in the process ATP synthesis, and this is the stroma inside. This is the ctDNA, means plastidome. It is circular naked, closed naked ring, and it is self-replicating and semi-autonomous. This is the ribosomes, 70S type ribosomes in the chloroplast, and this is the Frit membrane, also called the stroma lamellae or integrana lamellae. It helps in the transport, and ribosome helps in the production of synthesis of protein. Move to the next.